and I'd like to welcome you all to our fourth session of RVCOG overview classes. Today's focus is again from our Natural Resources Department, focusing on water quality programs and the Clean Water Act. And once again, we would like to welcome Amy from our Natural Resources Department. And Amy, I'll turn it over to you.
Okay, well, thank you all for joining in for this conversation today. I know you heard from me last week talking about the cleanup efforts that take place in both the Bear Creek watershed and also along the Rogue River. Once again, my name is Amy Sedlecki, and I'm the Natural Resources Technician for the Rogue Valley Council of Governments. And before I begin, I usually like to give a little personal tidbit just to open things up. So I have grown up in the Rogue Valley. My first job was actually at a scuba shop in Grants Pass. Still work there part-time. And when I learned how to scuba dive, and I was in the Illinois River and kind of seeing some effects of what people can do to our natural resources, leaving trash, polluting, it really got me to go in a direction, an environmental direction. And so I worked there, and I ended up going up to Portland, and I went to school up there, working on my environmental studies bachelor's degree. And I found myself in an internship back in the Rogue Valley, working for one of our partners, actually. I got to go out and collect stormwater samples for the summer and take those samples back to a lab and test them for a bunch of parameters that would indicate if that stormwater outfall was carrying polluted water into our streams. And I came out of that internship with a passion. And I knew that it was a temporary internship, but that if there was some way I was going to get back down here and live where I wanted to live and do what I wanted to do, and that was water quality monitoring. So I think this is really a perfect topic for me to present on. I might be a little quick on the Clean Water Act slides, just kind of get that information to you pretty quickly and jump right into the fun, the most fun part of my job. So probably going to be pretty passionate today. So let's go ahead and get started. So what are we going to be talking about? Today we're going to be focusing on some basics of the Clean Water Act. And that section 303D that you see there on the slide, that's part of the Clean Water Act. So I'll give a little bit more information on that, as well as the concept of a total maximum daily load. I'll go into Bear Creek's total maximum daily load, or TMDL, and then I'll just get to show you a bunch of pictures and tell you all about all of RVCOG's monitoring field runs that I get to go out on and collect samples for. So back in 1948 until about 1972, the Clean Water Act was known as the Federal Water Pollution Control Act. That's a bit more of a mouthful than the Clean Water. Actually, before I even continue, I want to make sure that you're seeing this next slide that says History of the Clean Water Act. Okay.
Yes.
Great. So the Clean Water Act gives a structure for regulating pollutant discharges. It gives the EPA authority to set water quality standards for all contaminants in surface waters. And it makes it illegal for people to just dump whatever they want into waterways and storm drains. The Clean Water Act funded the construction of wastewater treatment plants, and it also recognized the need to address problems associated with non-point source pollution. When I say non-point source pollution, there's really no way to pinpoint where that pollution is coming from. So we could get this, like what we've been seeing, a huge rain event, and have a bunch of contaminants come into our creeks and not really be able to pinpoint exactly where those contaminants are coming from, because that rain's just carrying all of that into the waterway. You can't really blame one specific person or one specific industry in that case, because you just got it all coming from different directions. So that's what I mean by non-point. Point sources are storm drains. So all of that is flowing into a storm drain, and that one point or stormwater outfall is a point source. So Section 303D is a list of water bodies that don't meet water quality standards. So the Clean Water Act, under that act, there's this Section 303D. Oregon's Department of Environmental Quality every 2 years assesses all of the water bodies in the state of Oregon. They identify which ones need to be placed on that 303D list because they're not meeting water quality, such as they're not meeting standards for contact recreation. If you go out and you want to swim in this certain creek, there's too much pollutants in it for you to safely swim there. That's what I mean by placed on that list. It's not meeting water quality standards. Now, not all creeks have they don't all have to meet that contact recreation standard. There are creeks that need to meet a standard to be fishable and consumable. So we've got certain creeks that you can draw water from. So it's just a variety of different water quality standards. And so for Bear Creek, those water quality standards are mostly contact recreation. Not a lot of people are going out there and swimming in it, but it would be pretty cool to one day be able to say, hey, this is a local resource and we can go out and wade in it. And so that's kind of the goal here. Bear Creek is one of many waterways in Oregon that's been placed on that 303D list. I'll go into a little bit more detail on that. So you can see in the picture here, there's a bunch of purple lines. Those are the waterways that are currently on that 303D list. So although it looks like most of our waterways are impaired or they're suffering, it says here, for example, DEQ assessed data for 25 parameters on a stream. And so even if just one parameter is out of line, then it's going to be flagged, and that's why it's shown on the map. So it's really not as bad as it looks. And going further, once a creek or a waterway is placed on that Section 303D list, it is given a total maximum daily load of that certain pollutant that can go in the creek. Further, TMDL is the calculation of the maximum amount of a pollutant allowed to enter a water body, so that water body will continue to meet water quality standards. And this is just a nice schematic that simplifies pretty much what I've already mentioned. But we go out and we identify a stream that's polluted. The next step is to study that creek and figure out what the sources of pollution are and how much of a pollutant can go into that stream to maintain those water quality standards. Then we come up with an implementation plan that involves maybe going out and, let's say the pollutant is too much heat getting into the stream. And that sounds funny, but that is actually a parameter that is related to Bear Creek, is that its temperature, it can get too hot. And so to implement an implementation strategy is to go out and plant trees that will eventually be big enough to shade that waterway and provide a way to cool the water down. That's just one implementation strategy. And additionally, we're also monitoring that water. So going out and collecting samples, taking them to the lab, and determining if pollutants are there and at what values. So Bear Creek was placed on the 303D list back in November of 1987. So it's been listed for a pretty long time. Back in 1987, it was listed for many more parameters than it is now. So Bear Creek has improved. Currently, there are total maximum daily loads for bacteria, specifically E. coli, and temperature. And as the regional manager, RVCOG assists all the designated management agencies. That's cities of Central Point, Medford, Talent, Phoenix, Ashland, Jacksonville, and then Jackson County, as well as some other special districts like Rogue Valley Sewer Services, Jackson Soil and Water Conservation District. So we are the regional manager of the Bear Creek Implementation Plan. Part of that plan is water quality monitoring, various other activities. For example, restoration, which Greg went over a couple weeks ago. And he's also talked about education and outreach last week. So this week's conversation is surrounding monitoring. So for Bear Creek, the sources of bacteria are various, and there's not always a way to pinpoint what exactly is the issue. Sometimes it's just a combination of everything. Actually, in most cases, it's a combination of all of these. So in more severe cases, it's failing septic systems and cross-pipe misconnections. So what I mean by that is you could have a sewer pipe accidentally hooked up to the storm drain system, and that has happened before. There's just your urban stormwater, there's effects from agriculture, wild animals, pets, and then there's illegal dumping. Sources of temperature increases are solar radiation, and you don't have the trees to provide the shade. You've got just water use, people taking water out for irrigation purposes. And as far as wastewater treatment plants go, sometimes that water that's been treated that goes back into the creek is a higher temperature than the natural flow that's already in the creek. So the TMDL effectiveness monitoring field run is probably the biggest field run that I get to go out and do. This is done on a monthly basis, and I collect samples from 23 different sites, and this is all taking place in one day. You can see in the map on the left, there are green circles, and those are the TMDL sampling locations that I go to. So Central Point, Jacksonville, all the cities here in the Bear Creek watershed, I get to visit on that one day and collect samples for bacteria. I get a conductivity reading, a pH reading, and we want to stick in that 6.5 to 8.5 pH reading. And most of the time, pH is perfectly fine in that range. Sometimes in the summer, we get up to 8.9, 90. The reading for temperature. Collect samples for total phosphorus during the months between May and October. And the reason for that is that's irrigation season. So total phosphorus can come from sources like fertilizers, anything that's involved in agriculture. Not anything, but lots of things in agriculture. That's why we are monitoring for total phosphorus during that time period. And turbidity. And turbidity is essentially how cloudy is that water, how much sediment is in the water. Can you see through it, or is it just a brown color because it's got so much dirt or soil in it? And this is our field sheet that we use. You can see all the different sites are given a site number, and the E just stands for effectiveness. And not all of the sites have always been on this monitoring run. They've changed over the years, but as far as I know, these sites have been in place since 2015. You can see that there's 3 bottles columns So there's 3 different types of bottles that I collect a water sample in. I've got a total phosphorus bottle, a bacteria bottle, and a pH bottle. So in each site, I'm getting 3 samples. And in addition to taking samples, I'm using a temperature and conductivity probe as seen here. Got my turbidimeter here. And for the probe, I'm just unwinding the cable and throwing the probe in the water and just waiting a minute or 2 to get a reading on that. For the turbidimeter, I've got these different vials here that I rinse 3 times and fill and then place in this compartment here. And what happens is a light is passed through that tube or vial, and it comes up with a turbidity reading. So it's probably a little bit difficult to see in the picture here, but it says 5.21. That's pretty low. So that vial of water probably looks pretty clear. I've seen up to 180, and that's when you've got a lot of sediment in the water, and it makes it like brown color and not clear. You can't see through it. I also wanted to include what our field vehicle looks like. I've got my 4 coolers in here. All of the samples need to be placed on ice until I can get back to the lab to test the water samples. We've got our buckets here for bucket sampling. And so the 23 sites are split up, 7 being in Central Point, 2 in Jacksonville, 3 in Medford, 2 in Phoenix, 4 in Talent, and 5 in Ashland. And I don't just take bucket samples, I also can access the creek, both Bear Creek and also Jackson, Griffin, Ashland Creek, they're smaller creeks. Access those and get a grab sample. So once the samples have been collected, next is testing at the lab. You can see the top left photo there is a tray full of sample testing how much bacteria is in the water. So what happens is I take my sample, it's about 100 milliliters of creek water. I add essentially a food for bacteria to that water sample. Take it up, place it in this special tray that's got 49 large cells and 48 small cells. It goes into an incubator for 24 hours, and after that 24-hour period, you take a black light and shine it over the tray. And any of those cells that are glowing, like you can see here, that's a positive sample for E. coli. After that, you use a special table that's got your large cells across the top. It's a spreadsheet. And you've got your small samples on the other side, and you count, okay, there's 20 large cells and 0 small cells, and you match those up on the spreadsheet, and that gives you your E. coli count. For the total phosphorus samples, it's a little bit more complicated. It's about a 6-hour test, and you line up all the bottles in the order that they were collected, put 50 milliliters into these special flasks here, and after a variety of chemical, adding different chemicals and placing them into an autoclave that heats them up to a certain temperature. Then you can see in these photos here where my cursor is, some samples are a bluish color. So after all those chemical reactions and heating up that sample, those flasks with blue, the more blue, the more phosphorus is in that sample. Then you take a smaller sample from the flask and put it in what's called a spectrophotometer. And this will pass a light through the sample, just like the turbidimeter, to determine a total phosphorus value concentration. So it's a pretty neat test. It is pretty time consuming, but it's good to know how much phosphorus is in the creek, and we can figure out a way to prevent that runoff from getting in there, because we don't want to be seeing this blue. We want to be seeing more clear samples. In addition to the TMDL monitoring run, I also go out and sample storm drains. And this happens 3 times per year, once during the dry weather season, so that I like to target June, July, August. There's the first flush, which is basically just your first big rain event that's going to have a lot of runoff on the streets, taking anything that's been there over the summer into the storm drain and then into the creek. And then wet weather, which we would classify last week and this week as a wet weather run if we were to go out. During those monitoring field runs, I collect bacteria, biological oxygen demand, so that is how much oxygen is being taken up by a certain pollutant that's in the water. And that is calculated after 5 days. Conductivity, again, pH, temperature, total phosphorus, and that doesn't matter if it's April, that's technically out of the total phosphorus season, but for the storm drain run, I'm taking those samples no matter what. And then turbidity, of course. So the photos on the bottom are 4 different storm drain sampling sites that I visit. You've got your pretty typical concrete outfall here. You've got a culvert, and then you've got a PVC pipe. So just different examples of what they look like. They're not all the same. And then on the map here, those black circles are showing where the storm drain sites is. So again, they're all over the Bear Creek watershed. This is our field sheet. And if you're very detail oriented, you'll notice that the list starts with the number 17. That has been that way since I started, so I don't know why it's out of order. I assume it's because we changed 1, 2, 3 and switched them for these different locations and just kept it that way. I don't know. It's just one of the quirks of the job, but these are the different sites that I go out and visit. And then there's 4 bottles this time, so that's why there are 4 coolers in the field vehicle for the 4 different types of samples. All right, so the 3 photos on the top is kind of showing you first the water coming out of the outfall, and then taking a step back and noticing, oh wow, that water is a lot lighter colored than the rest of the creek. You can see that more in this photo. What's going on there? So there's clearly a difference between what's coming out of the pipe and what's already flowing in the creek. So that's a location that gets flagged. So we'll let that municipality know, hey, we saw this. We'll give them our data that we've collected after it's been analyzed, and they'll go out and do their own investigation of what might be going on, what might be getting into the. Another example is this one here. You can see, again, it's a lighter color compared to the rest of the water. That was just last week. I notified that municipality what was going on, and they're going to go out and do an investigation to see what might be getting into that storm drain. And this, unfortunately, just happened last week, as I'm sure you're all very well aware of, the Medford fuel fire. And this photo was taken from the Jackson Street Bridge, and you can see that although a boom was placed out there, that fuel, the sheen, is just getting right past that. So I sent that photo in to Greg, and he contacted DEQ, and I'm hopeful that the booms were either redone or something happened to make it so that the boom was doing the job correctly. I mentioned biological oxygen demand. This photo here is just using a probe, a special probe that's got like a not a turbine, but some sort of a fan on it that gets the water moving around in the container. And it takes a reading of what that oxygen demand was after 5 days of sitting in an incubator. So the lower the value on this test, that means that whatever is in the sample was eating away that oxygen, so likely could do some damage to any water, aquatic life. That pollutant could take oxygen out of the water and make it a very hostile living environment. So that's why we do that test, to see how are these storm drains affecting Bear Creek and other waterways that they're flowing into. Another field run that I get to go and do is called the Quality Assurance Quality Control, or QA/QC monitoring. And this is done in conjunction with Department of Environmental Quality staff. What they do is they take a few days, go around the whole state, and take samples from different waterways. So the point of this monitoring run is for me to meet up with them at 2 sites 6 times a year, and we compare our conductivity readings, our pH readings. We just compare our numbers to make sure that RVCOG's equipment is working properly. And so that's just done at 2 sites, one at Kirtland Road in Central Point, White City-ish area, and then one in Gold Hill on the Rogue River. So there's a Bear Creek sample and a Rogue River sample. And how DEQ takes their samples are with metal buckets, as you can see in this photo here. We actually were lucky enough to acquire one of those buckets, and it's been really great, and it's been an arm workout for sure. Going from those plastic buckets that we have to this metal bucket is quite a difference, but it is a more sterile way of collecting a water sample. And I just wanted to put these 4 photos together because I like to stand at the same location each time I go out there. And so these are from different months. Very slight changes. This one's a little bit more easy to see that there's a difference. And you can see the water's really low here. That was likely back in July or August. And then this one was in December. There's also the cold water refugia monitoring, or going out and placing temperature loggers in different locations on Bear Creek, Griffin Creek, Jackson Creek. We might do Lone Pine Creek this year, as long as it doesn't dry up too early. And just trying to identify locations where there could be good habitat for salmonids. And what we like to do is place these temperature loggers in something that'll camouflage them. So this is just like a type of pipe that one of our past employees created and brought in. And other times we just use cans, really whatever we can do to camouflage these things, because they're like $100 a pop. We use nuts to weigh them down so they'll sink to the bottom. And what these little temperature loggers can do is pretty amazing. You can set them by hooking it up to a computer to take a temperature reading every 30 seconds if you wanted to. You can set it up every 5 minutes. What we've done is these loggers will take a reading every 15 minutes of temperature. And at the end of the season, so they're put in June and I take them out in October, I'll hook them back up to the computer and it will display a graph of how that temperature changed for those several months. We're working with the Rogue River Watershed Council and Oregon Fish and Wildlife and some other partners on this. So we all kind of collaborate on when we're going to go out, where we're going to place them, and work together, all for the sake of salmon. You can see in this picture here on the bottom center. We also have watershed patrols that we go out and do in the White City area, and this is a Rogue Drinking Water Partnership project. We're identifying areas where there could be pollution entering the storm drain system as well as directly into the Rogue River. We're trying to catch these any areas upstream of Medford's drinking water intake. You can see some of these photos, like the one on the left, there's a clear difference in the color of the creek there and what's been coming in. That's really light brown compared to more of a clearish color. You've got an oil sheen here in this wetland area. And then this is a industrial area that's just got, I don't know what, coming out of their pipe. So we just go out and let Medford know anything that we're seeing that we shouldn't be seeing and cause problems for drinking water. And then, of course, we've also got an Alameda fire monitoring field run that's done the same time as the storm drain runs or the TMDL runs. I just take 3 to 5 samples at locations that are upstream and downstream of where the Alameda fire started. You can see here there's a whole bunch of parameters that we're testing for. Now, why are we doing this? Why spend all this time and go out and collect samples and all? What are we going to do with the information? Well, after I'm done with the slideshow, I'll show you how the bacteria values have changed from month to month. First, what we like to do is identify those areas that could really use some help. So, for example, this is Larson Creek, and we identified that area as one needing some shade to provide colder temperatures. So this is my colleague Craig, and he's out planting some native shrubs along Larson Creek and also some tree species out there. We put in willow stakes, as if you can remember me talking about that last week. Those will eventually provide some shade and colder water for aquatic species. That's how we use water quality data is, okay, why are these temperatures so high? What can we do to lower those water temperatures? We also participate, like Greg mentioned last week, in lots of different education and outreach events, so we can let the public know what we're seeing and how they can help using the StreamSmart platform. And also to combat the issues with bacteria getting into the creeks, we can put in stormwater control measures such as other strategies like that. So collect the data, assess it, and decide what we need to do along our creeks to combat those issues. Just want to also mention we've got all these partners that help fund or provide in-kind services for us to go out and collect these samples and analyze them. So I want to thank our partners. And I'm not gonna end the slideshow just yet because I want to show you something else here. Great. Okay, is everybody seeing some graphs?
Yes.
Okay. All right, so for example, E14, Bear Creek at 9th Street in Medford. You can see that E. coli values were very high during the summer. They've come down. So as of March, they're below this red line here. The red line is showing the maximum amount that should be getting into that can get into the creek. So we're below that now. The summer is usually the worst. And once we get into the winter and spring, and it gets a lot better. So that's just one example of how bacteria levels change throughout the year. Sometimes they are not so typical. sometimes we get a lot of rain that might put a lot of bacteria all at once into the creek, and I just happened to be out there that day collecting, so it seems maybe a bit worse than it really is. So that's not really a typical looking graph there. So we just track how these bacteria trends look throughout the year. Also, phosphorus trends. And because total phosphorus isn't collected every single month, you're not ours. Again, that's just collected May through October. And so we're also watching those total phosphorus trends change throughout the year. And taking the data that we find and just finding averages, maximum, minimum, median range, just all those basic statistics. And I'm going to go back to the slideshow now. And hopefully move forward. Okay, so I know I moved through that pretty quickly. If you have any questions, I'm here for them. So bring them on.
Anne, go ahead with your question.
Oh, I've got some questions and some comments. Uh, one of this is like from a couple of OLLI classes, kind of get some hints about water, even peripherally with some of the classes that I'm in. For example, I just came from a class on community journalism where one of the students had done an article about Beach Creek, which I had no idea what Beach Creek was. But anyway, apparently there's going to be a big housing development around that area, 52 homes, and Beach Creek, I guess, connects with either Ashland Creek or I think it's Bear Creek, um, and with the disturbance of the earth and all that kind of stuff. The debris going into Bear Creek is one of the concerns about that project. So I mean, if you didn't know about it, it's kind of interesting because I didn't know about it and I live like across the street from where the development's going in. Um, anyway, and then another class I took where there was some discussion of the fact that the water and sewer pipes are very close together. And of course, sometimes there's some porous kind of, I guess, potential for contamination of the water pipe by the sewer pipe. And one of the methods that's used to try to avoid that is by pumping. I don't know whether it's the water or the sewer, but anyway, with the pumping action, it keeps the flow such that it might not seep between one or the other. And what I learned is that that's all dependent on diesel fuel at the pumping station, and if there were some kind of an event where that diesel weren't available because they just get it at the gas station, then that would be there's no like diesel depot in Ashland, so there would be an issue with contamination. So that was kind of interesting. So just some things that I discovered about Ashland. I guess the other thing is the potential. This is a mycology thing. So the potential for fungi to do remediation of things like that oil contamination. So I don't know if there's any, I mean, the boom, of course, is very helpful in, having the oil spill progress down the creek. But in terms of cleanup, there is that opportunity for fungi to do some of that, which you probably know all about. Anyway, that's my little excerpts from my OLLI courses.
Thank you.
Amy, I have something I thought maybe you could share with the group is where is this great lab with all this neat equipment? Because I know that RVCOG does not have such lab with all that neat stuff.
Well, we have a really awesome partnership with the City of Medford wastewater reclamation facility. They let us come in and use a lot of their equipment, and we have some of our own too, but they provide the facility, and they've been really great partners. I know sometimes I go in and I failed to email them that I'm coming by, and every time I go in, they say, "Oh, hi, Amy, how's it going?" Like it's really nice to work with them, and they're so helpful. So yeah, the City of Medford is a great partner in.
All right, thanks, Amy. Anne, go right ahead.
I'll tell you, I don't want to hog up all the discussion, but I do have some more things. One of them, and this is also kind of an OLLI thing, we had John Kroger, who was the Attorney General, come down for a, I guess, lecture some years ago, and he talked about the fact that the fines for dumping into the waterways, and of course, and he was thinking more, I think, the Columbia or whatever. But anyway, the fines, he said, are so trivial that for a dumper, it's more cost effective to dump than it is to pay the fine. So his thing was, let's just make it painful for them. So I think they've, I don't know whether that's statewide, but I know he was eager to go after some of the big offenders, and I don't know whether that's the fines have down here in our neck of the woods, whether they've gotten more of a deterrent than used to exist, and I don't know. So that's kind of a question, or and maybe you don't know about the penalties for that, but anyway, and I guess the other thing I noticed is that I didn't see SOU as a partner on your slide, and of course, the university touts their sustainability and environmental and all, that program. So I kind of wondered at that, um, why they might not be involved or whatever.
That photo that I showed with partners listed is a little outdated. SOU is a partner. I know that part of the Alameda fire monitoring, we do have SOU students come out and calibrate the data sonde. It's just the structure that collects continuous temperature, turbidity, just different parameters. And so they come out and calibrate that every month. I know that we've got other affiliations with SOU, but that's one that I know specifically. So yes, they are a partner. As far as the fines go, I'm not going to give any details on it, but we did have an opportunity to benefit from an industrial source dumping into Bear Creek, and how we were able to, and it's kind of weird to say benefit, but for the Bear Creek Stewardship Day cleanups, we used that fine to pay for a bunch of, just supplies for the cleanup. So they got fined a pretty hefty amount, but that money was able to go back toward cleaning up Bear Creek.
Great.
I don't know if that really answers your question, but just a little tidbit.
I'd just like to know they're getting their hands, more than a hand slap. Yeah. Well, actually, and speaking of the Bear Creek cleanup, again, for my community journalism class, you had to write stories. I mean, we had homework. Anyway, so I did one on Earth Day and talked about the 4 events that are happening on the 22nd to the 24th, which includes the Bear Creek Stewards project. So thanks for this class. I wouldn't have known about that otherwise. So.
Well, thank you.
Great. Yeah, I am pleased to report. I know, they don't always share the structure of the fines, and some of them are dependent on the level of the pollutant and the area it covered. But I am pleased to share, as Amy said, that one of those large fines that was for a local issue, the fine was funneled back locally. So, sometimes you wonder, well, when I get a speeding ticket and I pay for that, where does that money really go? So in this case, it's very encouraging that although, a lot of these fines are coming through maybe state or even federal agencies, it was really good to see that funneled back through. Some of them go, in this case, one came through our organization. We spearheaded that collaboration of cleanup. But there are also other times it may go directly to another partner agency that is, depending on the area, specific region. So it's nice to see that. I also think, we're all well aware of some issues and struggles we've had here, specifically in Southern Oregon, with the marijuana and hemp grows. So they have had a huge impact on water. So not only water rights and water theft, but also in the water quality of, again, what Amy's talking about and looking at those phosphorus levels, because just the level of pollutants that are going back into our waterways that are washing off of the marijuana and hemp grows. So those have all been issues. Again, sometimes it takes a while to kind of direct a response to these things. So it did take a little bit, but the state has recognized these specific struggles that we're having down here in Southern Oregon, thanks to many of our Jackson and Josephine County commissioners and partners and elected officials, representatives in the House and the Senate. So there is some funding that specifically came down here to Jackson, Josephine counties, not only to increase law enforcement abilities to, actually go out and enforce the rules and regulations, but also some funds towards trying to manage what this is doing to our area. And I know we've all seen those fly-by-night grows that are there one day and gone the next, and in their wake is just an incredible amount of trash and garbage. And again, now that we get these heavy rains and they wash across those fields that no longer have the supporting plants to clean some of that runoff, and it's running into the streams. And so I know, as Amy pointed out, here's something we notice in this storm drain, and we have the jurisdiction go take a look. And unfortunately, what they may find is there's now a new barren field that the water's running across, and it is picking up a bunch of things that, were there used to be some weeds at least, or native grass or something that was helping to collect that stormwater. So really important. I know a lot of people think that these ecological and environmental issues, are somebody else's problem, but they're very much not. And even every one of us as we go about our day, and cleaning up after our pets is so key and so important. And yet every day you see people who just don't seem to care that their pet waste is over there for somebody to step in, or to wash into our natural environment. So I know that's one thing that some of the little goodies that Natural Resources gets to hand out. One of the things that they were able to receive were some of those little, dog bone shaped things that you put your pet pickup bags in, and you attach it to your leash, and off you go. So there are a lot of things that we can do just as reminders to people to be good stewards of our natural resources. And we have a very beautiful region down here, and it's wonderful that we have people like Amy and the other folks in our natural resources department who have a passion for trying to keep things the way they should be. So Thank you, Amy. And we'll thank Greg and Craig, who were also in some of your photos.
And we can all go out for Earth Day,
Yes.
pick up trash, but also the what do you call it? The Earth Day extravaganza is going to be at the farm this year. The Rogue Valley Earth Day people are discontinuing the event they had at Science Works. But the farm is right adjacent to Science Works, and so many of the activities that were done at the Rogue Valley Earth Day will be done right next door, and and I think they're going to have like 30 booths and food and music and everything. So it should be a good happening for people here in Ashland and some education, right?
Mhm. Yes. All right. Amy, how are signups going for the Stewardship Day?
I just keep getting emails coming through saying this person signed up. So I think it's going to be pretty busy all week just with new signups.
Great. Awesome.
I do see a question from Michael in the chat. With many sources of pollution, how do you determine what quota is to be applied to each source? And that's a pretty big question. And I think I can only take a stab at it. But depending on which parameter, so let's say total phosphorus, when I analyze my samples and I get a sample that has an amount greater than 0.08 milligrams per liter of phosphorus, that is exceeding the acceptable value. So we take that 0.08 milligrams per liter and multiply it somehow by the cubic feet per second or flow that's of Bear Creek. So it's the allowable amount times the flow that's occurring at that given point, and that gives us how much is going through. So somehow they figure out an amount that's acceptable that will still make it so that Bear Creek can meet its water quality standards. So I don't know if that answer suffices, but that's what I got.
Well, if I understand, what you're saying is that you find the fraction of phosphorus over whatever denominator it is, and then you apply that to the volume of outflow from each of the sources. So that assumes that everything is uniform, right? And one place could be putting out 5 times as much stuff, but you would be assuming that it's proportional to the entire amount of pollutant that you observe. Is that right?
I suppose it's just taking into account what I collect at that specific location, which could be just barely upstream from a major polluter that could be putting in a bunch. Yeah. So it's not a perfect system.
Okay, thanks.
All right.
Sam has,
Sam, I see your hand up.
Yes, I just wanted to add to what Anne Belagia said about Earth Day. I didn't hear her mention, and maybe everybody just knows, but the farm is the farm at SOU.
Yeah, thanks for the.
That was it.
It's on what? Yeah, it's on Walker, across from the whatever it is, Ashland Middle School, I guess. Yeah.
Yeah, I think that's it. Yeah.
There's sort of another thing that I learned about Oregon Garden, which I never thought about that as having any kind of ecological value, but apparently there was a lot of water temperature issues being dumped into whatever river is adjacent to that, and the Oregon Garden was actually the part of the remediation. I guess it's sort of like a bioswale, but they actually created plants as stepping down into the river, so it did the cooling thing, and then they got themselves an actual, I don't know, tourist attraction. So kind of a win-win for that solution.
All right, Mary, 1 second. We have a question from Norman in the chat and then we'll get to you. So Norman is asking, do we ever check for more exotic pollutants like antibiotics or Roundup or other drugs? Or are we just focused on the core ones that you shared?
As far as Roundup goes, there's a pesticide stewardship partnership that I believe RVCOG has been involved in the past. We're not currently involved in that effort, but there is a PSP effort that goes out and collects samples to determine how much of Roundup or other similar pollutants like that are in waterways. So yes, that is a thing. As far as antibiotics or other drugs that might be coming from human sources or animal sources, I know several years ago, RVCOG took samples and had them analyzed for different human impacts like that, but we're not currently doing that. The investment in that and the outcome just wasn't worth it at the time.
Okay, thank you.
Thanks, Amy. All right, Mary, your question next.
Just want you to know the farm is across the street from the junior high on Walker. You can't miss it. It's after the railroad crossing.
All right, thank you, Mary. All right, any other questions or thoughts before we sign off for today?
It's been very informative.
Yes, it was. And thank you for another picture of Alex at the end. You should have left
I
him
think
up.
it might. I think it's going to be a theme
Yeah. Okay.
until someone tells me to stop.
No, we'll keep going. He'll be 14, 15 years old, and there will be Alex at the end of your slideshow. So I want to thank you all for showing up again for the 4th class. And just a reminder, next week's class, session 5, we are going to switch gears and we are moving on to Food and Friends Senior Nutrition. So we're going to talk about our senior nutrition program. They run our congregate meal sites in Jackson and Josephine County, as well as all of the Meals on Wheels home deliveries. So we will be meeting up with John and or Christy next week for our presenters. And I thank you again for joining.
Thank you.
All right. Bye. Enjoy the rest of your afternoon, everyone.
Thank you.